Hello, and welcome back to AP Biology, Semester 2. We are in the big idea 2, free energy. And today we are talking about the cell. And we're in Chapter 6, Section 4, where the endomembrane system regulates protein traffic and performs metabolic functions in the cell. Many of the different membranes of the eukaryotic cell are part of an endomembrane system, which carries out a variety of tasks in the cell. These tasks include synthesis of proteins and their transport into membranes and organelles or out of the cell, metabolism and movement of lipids, and detoxification of poisons. The membranes of this system are related either through direct physical continuity or by the transfer of membrane segments as tiny vesicles, which are sacs made of membrane. Despite these relationships, the various membranes are not identical in structure and function. Moreover, the thickness, molecular composition, and types of chemical reactions carried out in a given membrane are not fixed, but may be modified several times during the membrane's life. The endomembrane system includes the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, lysosomes, various kinds of vacuoles, and the plasma membrane. Not actually an endomembrane in physical location, but nevertheless related to the endoplasmic reticulum and other internal membranes. Having already discussed the nuclear envelope, we will now focus on the endoplasmic reticulum and the other endomembranes to which the endoplasmic reticulum gives rise. Okay. The endoplasmic reticulum is such an extensive network of membranes that it accounts for more than half the total membrane in many eukaryotic cells. The word endoplasmic means within the cytoplasm, and reticulum is Latin for little net. The ER consists of a network of membranous tubules and sacs called cisternae, from the Latin cisterna, a reservoir for a liquid. The ER membrane separates the internal compartment of the ER, called the ER lumen, or the cavity, or cisternal space, from the cytosol. And because the ER membrane is continuous with the nuclear envelope, the space between the two membranes of the envelope is continuous with the lumen of the ER. There are two distinct, though connected, regions of the ER that differ in structure and function: smooth ER and rough ER. Smooth ER is so named because its outer surface lacks ribosomes. Rough ER has ribosomes on the outer surface of the membrane and thus appears rough through the electron microscope. As already mentioned, ribosomes are also attached to the cytoplasmic side of the nuclear envelope's outer membrane, which is continuous with rough ER. Now, let's talk about the functions of the smooth ER. The smooth ER functions in diverse metabolic processes which vary with cell type. These processes include synthesis of lipids, metabolism of carbohydrates, and detoxification of drugs and poisons. Enzymes of the smooth ER are important in the synthesis of lipids, including oils, phospholipids, and steroids. Among the steroids produced by the smooth ER in an animal cell are the sex hormones of vertebrates and the various steroid hormones secreted by the adrenal glands. The cells that synthesize and secrete these hormones in the testes and ovaries, for example, are rich in smooth ER, a structural feature that fits the function of these cells. Other enzymes of the smooth ER help detoxify drugs and poisons, especially in liver cells. Detoxification usually involves adding hydroxyl groups to drug molecules, making them more soluble and easier to flush from the body. The sedative, phenobarbital, and other barbiturates are examples of drugs metabolized in this manner by smooth ER in liver cells. In fact, barbiturates, alcohol, and many other drugs induce the proliferation of smooth ER and its associated detoxification enzymes, thus increasing the rate of detoxification. This, in turn, increases tolerance to the drugs, meaning that higher doses are required to achieve a particular effect, such as sedation. Also, because some of the detoxification enzymes have relatively broad action, the proliferation of smooth ER in response to one drug can increase tolerance to other drugs as well. Barbiturate abuse, for example, can decrease the effectiveness of certain antibiotics and other useful drugs. The smooth ER also stores calcium ions. In muscle cells, for example, a specialized smooth ER membrane pumps calcium ions from the cytosol into the ER lumen. When a muscle cell is stimulated by a nerve impulse, calcium ions rush back across the ER membrane into the cytosol and trigger contraction of the muscle cell. In other cell types, calcium ion release from the smooth ER triggers different responses. All right. Now we're going to talk about the functions of the rough ER. Many types of cells secrete proteins produced by ribosomes attached to rough ER. For example, certain pancreatic cells synthesize the protein insulin on the ER and secrete this hormone into the bloodstream. As a polypeptide chain grows from a bound ribosome, it is threaded into the ER lumen through a pore formed by a protein complex in the ER membrane. As the new protein enters the ER lumen, it folds into its native shape. Most secretory proteins are glycoproteins, proteins that have carbohydrates covalently bonded to them. The carbohydrates are attached to the proteins in the ER by specialized molecules built into the ER membrane. And here I'm going to move this over so that you can get these definitions. All right. Hopefully that'll work most part. All right. Just follow along and you'll be okay. You can rewind as much as you need to get the notes that you need. You'll be fine. Okay? After secretory proteins are formed, the ER membrane keeps them separate from proteins that are produced by free ribosomes and will remain in the cytosol. Secretory proteins depart from the ER wrapped in the membranes of vesicles that build like bubbles from a specialized region called transitional ER. Vesicles in transit from one part of the cell to another are called transport vesicles. We will discuss their fate shortly. In addition to making secretory proteins, rough ER is a membrane factory for the cell. It grows in place by adding membrane proteins and phospholipids to its own membrane. As polypeptides destined to be membrane proteins grow from ribosomes, they are inserted into the ER membrane itself and are anchored there by their hydrophobic portions. The rough ER also makes its own membrane phospholipids. Enzymes built into the ER membrane assemble phospholipids from precursors in the cytosol. The ER membrane expands and is transformed in the form of transport vesicles to other components of the endomembrane system. Okay, now we are going to go on to the Golgi apparatus, and how it functions. After leaving the ER, many transport vesicles travel to the Golgi apparatus. We can think of the Golgi as a center of manufacturing, warehousing, sorting, and shipping. Here, products of the ER, such as proteins, are modified and stored and then sent to other destinations. Not surprisingly, the Golgi apparatus is especially extensive in cells specialized for secretion. The Golgi apparatus consists of flattened membranous sacs, cisternae, looking like a stack of pita bread. A cell may have many, even hundreds of these stacks. The membrane of each cisterna in a stack separates its internal space from the cytosol. Vesicles concentrated in the vicinity of the Golgi apparatus are engaged in the transfer of material between parts of the Golgi and other structures. A Golgi stack has a distinct structural polarity with membranes of cisternae on opposite sides of the stack, differing in thickness and molecular composition. The two poles of a Golgi stack are referred to as the cis face, and the trans face. These act respectively as the receiving and shipping departments of the Golgi apparatus. The cis face is usually located near the ER. Transport vesicles move material from the ER to the Golgi apparatus. A vesicle that buds from the ER can add its membrane and the contents of its lumen to the cis face by fusing with a Golgi membrane. The trans face gives rise to vesicles which pinch off and travel to other sites. Products of the ER are usually modified during their transit from the cis region to the trans region of the Golgi. For example, various Golgi enzymes modify the carbohydrate portions of glycoproteins. Carbohydrates are first added to proteins in the rough ER, often during the process of polypeptide synthesis. The carbohydrate on the resulting glycoprotein is then modified as it passes through the rest of the ER and the Golgi. The Golgi removes from sugar monomers and substitutes others, producing a large variety of carbohydrates. Membrane phospholipids may also be altered in the Golgi. In addition to its finishing work, the Golgi apparatus manufactures certain macromolecules by itself. Many polysaccharides secreted by cells are Golgi products, including pectins and certain other non-cellulose polysaccharides made by plant cells and incorporated along with cellulose into their cell walls. Cellulose is made by enzymes located within the plasma membrane, which directly deposit this polysaccharide on the outside surface. Like secretory proteins, non-protein Golgi products that will be secreted depart from the transface of the Golgi inside transport vesicles that eventually fuse with the plasma membrane. The Golgi manufactures and refines its products in stages with different cisternae containing unique teams of enzymes. Until recently, biologists viewed the Golgi as a static structure with products in various stages of processing transferred from one cisternae to the next by vesicles. While this may occur, recent research has given rise to a new model of the Golgi as a more dynamic structure. According to the model called the cisternal maturation model, the cisternae of the Golgi actually progress forward from the cis to the trans face of the Golgi, carrying and modifying their cargo as they move. Before a Golgi stack dispatches its products by building vesicles from the trans face It sorts these products and targets them for various parts of the cell. Molecular identification tags, such as phosphate groups added to the Golgi products, aid in sorting by acting like zip codes on mailing labels. Finally, transport vesicles budded from the Golgi may have external molecules on their membranes that recognize docking sites on the surface of specific organelles or on the plasma membrane, thus targeting the vesicles appropriately. Now we are going to, let's talk about lysosomes. And then after the lysosomes, we'll talk about the vacuoles, and then the endomembrane system, and then we will be done for today. All right? So, a lysosome is a membrane sac of hydrolytic enzymes that an animal cell uses to digest macromolecules. Lysosomal enzymes work best in the acidic environment found in lysosomes. If a lysosome breaks open or leaks in its contents, the released enzymes are not very active because the cytosol has a neutral pH. However, excessive leakage from a large number of lysosomes can destroy a cell by autodigestion. Hydrolytic enzymes and lysosomal membrane are made by rough ER and then transferred to the Golgi apparatus for further processing. At least some lysosomes probably arise by budding from the transphase of the Golgi apparatus. Proteins of the inner surface of the lysosomal membrane and the digestive enzymes themselves are thought to be spared from destruction by having three-dimensional shapes that protect vulnerable bonds from enzymatic attacks. All right. Okay, lysosomes carry out intracellular digestion in a variety of circumstances. Amoebas and many other protists eat by engulfing smaller organisms or other food particles, a process called phagocytosis. From the Greek, phagine, to eat, and kytos, vessel, referring here to the cell. The food vacuole formed in this way then fuses with a lysosome whose enzymes digest the food. Digestion products, including simple sugars, amino acids, and other monomers, pass into the cytosol and become nutrients for the cell. Some human cells also carry out phagocytosis. Among them are macrophages, a type of white blood cell that helps defend the body by engulfing and destroying bacteria and other invaders. Lysosomes also use their hydrolytic enzymes to recycle the cell's own organic material, a process called autophagy. During autophagy, a damaged organelle or small amount of cytosol becomes surrounded by a double membrane, which is of unknown origin, and a lysosome fuses with the outer membrane of this vesicle. The lysosome's enzymes dismantle the enclosed material, and the organic monomers are returned to the cytosol for reuse. With the help of lysosomes, the cell continually renews itself. A human liver, for example, recycles half of its macromolecules each week. Okay, now we're going to talk about the vacuoles. Vacuoles are membrane-bounded vesicles whose functions vary in different kinds of cells. Food vacuoles, formed by phagocytosis, have already been mentioned. Many freshwater protists have contractile vacuoles that pump excess water out of the cell, thereby maintaining a suitable concentration of ions and molecules inside the cell. In plants and fungi, which lack lysosomes, vacuoles carry out hydrolysis. However, they play other roles as well. Mature plant cells generally contain a large central vacuole. The central vacuole develops by the coalescence of smaller vacuoles themselves derived from the endoplasmic reticulum and Golgi apparatus. The vacuole is thus an integral part of a plant cell's endomembrane system. Like all cellular membranes, the vacuolar membrane is selective in transporting solutes. As a result, the solution inside the central vacuole, called cell sap, differs in composition from the cytosol. All right, we are almost done. The endomembrane system. It's a review. All right. The flow of membrane lipids and proteins through the various organelles as the membrane moves from the ER to the Golgi and then elsewhere, its molecular composition and metabolic functions are modified along with those of its contents. The endomembrane system is a complex and dynamic player in the cell's compartmental organization. We'll continue our tour of the cell with some membranous organelles that are not closely related to the endomembrane system, but play crucial roles in the energy transformation carried out by cells. And until next time, ta-ta!